ये दिखने के लिए वो यहाँ पे है। Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and we are now live session is on, so you can ask me the questions if you have any queries or if you would like to discuss any topic from genetic engineering and biotechnology. I prefer that you discuss the lectures whatever we have not discussed before, so that. But other than that, also if you have anything which you have seen the videos until now, you can discuss me anything, or you can discuss some other questions also. So Pallavi is asking about how the host DNA is protected from the tissue endonuclease activity. So you know that every DNA is methylated. So that methylation pattern is uh, is actually protecting these DNA from the restriction endonuclease of the host itself, but they are not protected from the restrictionizations of the another organism. So it's important, like, so if host DNA is getting protected because you have the methylase system, and that methylase system is going to methylate the crucial a nucleotides, and that is actually going to not allow the restriction enzyme to bind to form the closed complex, and once they will not be able to form the closed complex, they will not be able to cut the DNA. I hope it's clear. If it's not clear, you can again ask me the question again. Uh, as far as the question pattern is concerned, it is actually going to be the similar kind of pattern what you have just seen for the assignments. So it is going to be multiple question type or multiple MCQ type, and I prefer to ask some questions from theory and some questions from the the analytical type of questions, and some questions which are like application oriented questions. So it is going to be a mixture of all the three different aspects. Like we will try to judge the theory part, we are going to judge the application part, and we will also try to see how whether you can be able to be able to design the strategies as per the question requirements. So I urge the students to ask me the questions as soon as possible, because this session is very limited and it's a very short duration. So you should take the full opportunity to ask me anything you are, you don't understand from the course or in general also. So Bhagirat is asking about the role of junk DNA in the active gene expressions. So there is no precise role known for the junk DNA in the gene expressions. In fact, why that junk DNA is present, that is also not known. So, and then Divya is asking, how can we utilize the knowledge of genetic engineering for from entrepreneurial point of view? See, the course genetic engineering theory and application is giving you complete information about how you can start from the scratch and how you can be able to develop a product. As far as we are talking about the product which is proteinaceous in nature, whether you can use that to generate the antibodies, whether you are going to use that for a therapeutic proteins and so on. And some I have also discussed some of the applications also. So if you have a very clear idea about the whole protocol and whole procedure and the flow of informations, then probably you can use that to establish the you can use that information to develop some entrepreneurships and all those kind of things. In fact, if you have a clear idea, then probably it will be easier for you to establish the things rather than you have a very little or no idea about all these aspects. Uh, in my opinion, I think the Swagata is asking uh, that the assignment is very tough. See, assignments are not very tough if you go through with the content and the video what we have discussed. Everything is from the content only. It may in some few courses, few questions. It is not very straight, like it's not theory based. Sometimes it is application or the the uh, like more related to uh, how you can use that information. So sometimes it is analytical as well, but that is the fun of attempting these assignments, because if you just ask the theory part, most of the theory part is and if all the assignments and the questions are going to be based on the theory, then it is going to be of no fun to attempt those questions. Then Purnabrata is asking how covalent chromatography technique performance, how covalent chromatography technique performance irreversible bonding along with matrix. See, the covalent chromatography is utilizing a sulfhydryl linkage through which it is actually binding the protein of your interest, and it is also utilizing that to bind the functional group to the matrix. And if you can elaborate your question, that is probably going to help me to understand what you want to, what is your query and what you want to know actually. uh, see, the MOOCs course has nothing to do with the MSc Biotechnology internship program. It is just a way to give you the knowledge as well as the certificate associated with this. So IIT Guwahati, you welcome to the IIT Guwahati, but that will be the acceptance of the individual faculty, including myself, whether they will having a space for a the summer intern or winter intern or so on. So you should explore in different labs in biotechnology, and if they will be having a space, they will actually going to accept you as a research intern. Right now, the problem is that because of the COVID-19, majority of the people are either see our own BTech students are outside the campus, so how we can actually accept someone which is outside the campus or outside the city also. So once this pandemic situation is going to be improved, probably there will be an opening for you, and then we have a detailed plan, like detailed program, how we actually select the people in the internship and how we can actually take them to our lab. So you are highly welcome to the IIT Guwahati, but you should contact the individual faculties. I think I have already discussed about the exam pattern, what Swagata is asking. I cannot elaborate much about the questions, but I'll tell you that approximately 30 to 40% question would be an exact repeat of the question what I am asking in the assignments. Apart from that, there will be some questions which are going to be of analytical type. There will be few questions which are going to be theory based, and so on. So, exam pattern is going to be exactly the same as what you see in these tiny assignments, like where I am asking few questions of theory based, few questions of assignments like analytical type, and so on. But since it is exam, it's going to be little more tough compared to what you get in the assignment, because assignment you're only covering very small portion, like 2 to 3 lectures, whereas in the exam we are have to cover approximately 30 to 37 lectures. So that time the content is going to be more bigger, and that's how the questions are also going to be more complicated. People should be very fast in asking me the questions. We only have now 20 minutes left to wind up this session. So you should be very quick in asking the questions. Uh, in the exam, I think I am going to ask the 100 questions, and I think you will going to have the 3 hours to attempt those questions, and it will be of MCQ type. So, on like as it like simply like what we are doing the assignments. So you can imagine that the content what we are going to ask in the exam is like a equivalent to the 10 assignments what you have just done, right? So uh, something like that. But the questions are, as I said, some questions are almost 30 to 30% questions are going to be just exact repeat of what we are asking in the assignment. So those questions are going to be easy because you know the answers. Out of 12, I think it is the 8 assignments what you have to submit, right, Rekha? Rekha, 8 assignments, right? Out of 12, we have to submit 8 assignments. Okay, anyway, I think 8 assignments. Yeah, so Divya is correct. So Divya has only said, right? 8 assignments where you have to get 40% marks, and then you also have to get 40% or more in the in some exam. Then only you are eligible for getting the certificate. Yeah, if you submit all the 12, I think you will have a very high chances of getting more and more marks, because what we are going to judge is the 8 best. So suppose you got in on any of your assignment you have done something which is not up to the mark, then we will not going to take up those if the number is more than 8. So Divya is correct, very much correct. If you submit more assignments, there are more likely chances that your final score is going to be on a higher side. I think I really don't know about the centers. I think if you want to have any questions and queries related to the centers, you should ask the NPTEL office. You can reach to NPTEL IIT Guwahati or NPTEL office at IIT Madras, and you can ask them where is going to be their center, right? And I'm sure when you have opted for the exam, and when you opted for, and you when you paid the the when you have paid the uh, fees also, that time probably they may have asked you to choose the centers, or they will assign the centers very close to your place. So don't worry about the centers actually, but as I said, if you have any centers, you should, if you have any query related to centers, exam centers, you should contact the NPTEL IIT Guwahati office or NPTEL IIT Madras office. The email address is nptel@iitgac.in, and that you should write your queries, and they will actually going to answer. If you missed more than 5 assignments, you will not be eligible for getting the certificates, irrespective of how much marks you get in in the final exams. So Abhishek, I think uh, you can sit in the exam if you have already filled the form and you have already paid the fees, so that you will know the pattern and other things. But if you have skipped 7 assignments and you have not been able to submit, then you are not eligible for certificate. Divya, I think this course is very much designed in such a way that you will get the major section covered in this course. Like you, except from this course, if you study this particular uh, MOOCs course, apart from this course, you might have to study the biochemical engineering, and you also have to study the computational biology and the quantitative biology. If, apart from that, everything is covered in this course, and little bit immunology you might have to study. So, as far as the biotechnology is concerned, most of the topics I have tried to cover, but in the biotechnology gate exams, you also have the components from the immunology. You have the components from the biochemical engineering, and you also have the component from the general aptitude and the computational biology. So those topics you might have to take up separately. But other than that, I think this course is designed keeping in mind that some of you might be preparing for the gate exams. In assignment 12, what the Swagata is asking, so in the assignment 12, it's going to be the questions from all the previous assignments. Like it is going to have the questions where, because this is the last week, so we are actually going to revise the content. So when we are going to do the revision, the questions are also, the assignment is also going to be like from the previous. The questions are going to be like from the content whatever we have discussed from the module 1 to module 11. So assignment 12 is actually going to be a summary of everything. And what Divya is saying that why not I should upload the previous year's question paper. So I think I have to ask the NPTEL whether I can be whether it is allowed to upload. If they will allow us, then probably I'll upload and then you can be able to practice. So the session is almost more than 50% is done. If you have any more questions, you should ask me because this is an opportunity when you actually can ask me anything you want. Whether those questions are silly questions or whether those questions are not related to this course. Don't hesitate in asking the questions because if you hesitate, then you will miss this opportunity. So I strongly request you all that you should ask me the questions, whether it is in your opinion, whether it will be like silly questions or whether it will be very not related to this course, doesn't matter because I will be happy to answer if at all I will be in a position to answer. Uh, so I think I really don't know this answer. What is the reason of virulence in corona? Because I am not a very expert virologist. Uh, but as far I know that the corona is you know is is a simple viral disease. It's not very difficult to manage. What it is doing is it is only catching those people whose immune system is weak or vulnerable for these kind of infections. So there's nothing like weak or strong immune system. It is actually the immune system which should be appropriate to clear the infections. So what happen is if the immune system cannot clear the infection, and it will be keep triggering the reactions and keep trying to recover from these infections, then in those cases it is actually reduced, because anywhere if there will be a battle going on, that battle is actually going to destroy the land. So that is what exactly is happening in this case also. Because the coronavirus is infecting the lungs, the lung is the battleground. So if the lungs are getting, and then that lungs alveoli are the single thin membrane cells. So, and that is required for oxygen to be diffused into the cell, and that's how we can actually take up those oxygen from the environment. So if that there will be a battle going on, then that battle is actually going to destroy these alveoli, which are actually going to be work like a land, right? So once there will be this uh, land is destroyed, then the persons will not be able to breathe properly. And apart from that, then it is also going to start creating troubles in other organs like lung, like livers and kidney and heart and all that. Uh, so that is the reason. But as I said, I don't know the best cause of the virulence in corona. Uh, Probably you should, as far as the process to get the article published in national and international journals. Uh, I think what you could do is you should first choose a very interesting research topic, then do a literature search, see what kind of work is already being done. If you are asking about the review article, then your topic of reviewing something should be of a very high importance. Then only the people will be excited to read that particular article, and then only they will be ready to accept. So, for example, right now, if you would like to write a review article on the corona and all that kind of thing, that could be a very very interesting topic. And then you choose the appropriate journals or appropriate textbooks or appropriate place where you would like to submit. Then you go through with these journals, introduction to authors and so on. So this is just a very long procedure, and I think that cannot be explained in a couple of minutes. So if you would be interested to understand the process of article publications and writing process, I think there are a couple of good MOOCs courses are available on that. So you can go through the content of those courses, and that actually is going to give you the much better idea. So Purnarvata is asking how many fragments would be generated if you treat the following peptide with hydroxylamine. If you So I'll tell you the way how to do it, and then you can do it yourself, and then you can match the answers with the given right answer in the assignment itself. See, I have already discussed about the recognition sequence of the hydroxylamine, and then if you go with that recognition sequence, you will know after that particular amino acid, after that particular recognition site, it is actually going to cut. So if you cut this peptide, for example, if we have only one recognition site, then also you are going to get two peptides. If it is going to be three, then it is going to give you the 4 sequence. So as many times it cuts, you are actually going to get the peptide number 1 more to that. So just go through with the recognition sites, what is given in the video lectures, and then do it yourself, and that is actually going to give you the answers. Do we have any more questions? Because we have another 5 minutes to close this session. So this is the last minute. If you don't have any more questions, probably we'll wind up the live sessions. So I request you, those who are watching, should ask the questions, whatever is query, whether it is a silly query or whether a very good query or related to this course or related to your, uh, you know, uh, if you want to discuss something related to how to pursue and all that, that can be also can be discussed, but not on a priority basis. First we are going to discuss the content what we have in this course itself, and if the time will left, then we're going to discuss about the how to go ahead and do all this kind of thing. So if you have any more queries, we have another minute or so to go with this live sessions. Otherwise, we'll wind up. So I think Divya, if you want to go to the good universities like Harvard or Stanford or Yale, and any all those kind of good universities, it depends what you want to do after that. But apart from that, if you want to go to these universities, you have to work really hard. You have to not only clear the GRE and TOEFL and all those kinds of exams, but you also have to, what I suggest is you should actually get associated with some labs. Maybe if it is not possible to go to a, to some labs in CSR labs or some labs which is far away from your hometown. Better to be associated with any labs in your hometown itself, whether it will be on a voluntarily basis or whether you can take up some job. That's very subjective, but uh, the idea is if you go with some research experience, if you go with some kind of technical skills, then these universities are actually going to shapen those study and your experience, and then only you will be able to excel in these universities. Apart from that, as far I know, that the Harvard or Stanford type of universities not only look for the higher percentage or good GRE scores and all that, they also look for what kind of analytical capabilities you have. So you also should develop your analytical skills. Like you try to solve the questions like imaginary questions. I'm not saying that you can solve the real questions, because real question comes only when you start working in a laboratory. But apart from that, you also should publish one or two papers, because what they want to see is that you are actually having some publication in your bag. Then only your application becomes very strong for these kinds of universities. So if you want to go to Harvard and other kinds of places, you should publish something, even if it is a publication in a low impact factor or no impact factor journals, then also it is fine. But don't publish it in a journal which is like money oriented journal. Like you don't publish it in a open access journal which is not going to be uh, you know having any credit, because whatever you submit they will publish, because they are in more interested in money but rather than the content. But apart from that, like for example, current science is a very good place to publish. Journal of Biosciences is a very good place to publish. So if you publish even the like mini reviews or letter to editor and those kind of things. But if you want to do that, you have to study. You have to study a lot about the topics and the related content. I hope you understand. So I think we are winding up the sessions now, and good luck for your exams. And if you have any more queries, we are going to have the session 3 by the end of this month or early next month probably. So you can that time also you can ask me many more queries. Thank you.